Hello, and it's me, Miss Ellis. I'm back to talk to you a little bit about meiosis. So you finished the Pogles. You kind of got an introduction into what's happening. So I want to we're going to dip our toes into the process of meiosis and just kind of talk about the whole thing, and then we'll look at a little bit more details later. Before we begin, I want to show you this short 5-minute video from the Amoeba sisters. The numbers of chromosomes and chromatids and all that can be confusing, so we'll watch this quick video to kind of get an idea of what we're saying when we use this vocabulary.
If you have ever studied mitosis or meiosis, chances are the whole chromosome number thing, it's crossed your mind. I mean, how do you start out with a cell that has 46 chromosomes, and then it does mitosis, and then you end with two cells that each have 46 chromosomes? Or in meiosis, you end up with 4 cells that each have 23 chromosomes. It's like, here's a chromosome, there's a chromosome everywhere, a chromosome, but how? Well, this video is going to explain the chromosome numbers in these processes, but please watch our mitosis and meiosis video first so that you can understand the actual process first. And also, our video is talking about chromosome numbers in human cells, so please assume that for the entire video. And just a reminder, human body cells have 46 chromosomes, and human sex cells, also called gametes, they have 23 chromosomes. Now, chromosomes, as you know, they're made up of DNA and protein. The DNA is highly condensed, tightly wound. That's really important because it means that when the cells divide and they have to move that DNA over, it's easy to do that when the DNA has condensed into chromosomes. It makes DNA so portable and organized, which is great in processes like mitosis and meiosis because when you make new cells, you also have to get the DNA into those cells. This is a chromosome. This is also a chromosome. The difference is that the first one only had 1 chromatid, or copy, and the second one has 2 chromatids, or copies. The area in the middle of the chromosomes, this is called a centromere, and the centromere is where the spindle is going to attach. And that's kind of a big deal because that's how you're going to move the chromosomes during any division process. Generally, when you count chromosomes, you can count them by the number of centromeres present. Now, interphase is a step that happens one time before mitosis and meiosis can start. And in interphase, this is where we duplicate the chromosomes. So first we start with 46 of these chromosomes, which is how many chromosomes are found in human body cells. And as you can see in our little picture, they kind of look like little sticks. But after duplicating in interphase, we still have 46 chromosomes technically, but they have 2 chromatids. We call them sister chromatids, and they're held together at 1 centromere. See, as long as those chromatids are still attached to the centromere, we still count 46 chromosomes, but they're duplicated. And if they weren't attached like this, I mean, technically you could say there were 92 chromosomes. But right now in this picture, there are 46 chromosomes and 92 chromatids. Here's a little chart here because I love charts. So is there a time in mitosis when there are 92 chromosomes? Well, technically, you could argue that at anaphase, when the sister chromatids are separating and they're no longer attached, you could see in this picture there could be 92 chromosomes because they're not attached during that one phase. And that's why our chart has a little asterisk here. Although we still usually refer to them as chromatids during this process of cell division. So at the end of mitosis, those chromatids separate and you end up with 2 cells after mitosis that each have 46 chromosomes. Those chromosomes kind of look like little sticks in this picture. The 2 daughter cells, if you look, are identical to the starting cell in mitosis. Now let's take a look at meiosis. We have 46 chromosomes in the starting cell, and right now they each have 1 chromatid, just like with mitosis. And they duplicated interphase, and we still have 46 chromosomes now, and there are 92 sister chromatids, just like we started with in mitosis. But at the end of meiosis 1, you have 2 daughter cells, and they each contain 23 chromosomes with 46 chromatids. At the end of meiosis 2, you have 4 cells that each have 23 chromosomes, 23 chromatids. Just a reminder that in that clears it up a little bit for you. Now just remember that chromosome counting, it has a lot to do with the way we count chromosomes and this definition of chromatid. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, Amoeba Sisters, we will stay curious. All right, so on the text that you have, we're going to add some highlights and comments and annotations. So the process that produces haploid gametes is meiosis. BT dubs, I pronounce it meiosis because I had a Hispanic professor in college, and that's how he said it, so I picked that up. But you can say it either way. So meiosis is a type of cell division in which the number of chromosomes is reduced by half, and it only occurs in certain special cells. Those special cells are egg and sperm, which we call gametes. So this is only happening in one place in your body. Every other somatic or body cell undergoes mitosis, and the cells are clones. So it occurs in these gametes of organisms. During meiosis, homologous chromosomes separate, and the haploid cells that form have only one chromosome from each pair. Now I'm going to talk more about that homologous chromosome separation in just a little bit. The phases of meiosis 1. Meiosis 1 begins after DNA replication during interphase. In both meiosis 1 and 2, cells go through the same phases as mitosis. However, there are some important differences between meiosis 1 and mitosis, and that's what we're going to look at next. So meiosis 1, of course, starts with prophase 1. Same things that are happening in mitosis are happening here. The nuclear membrane breaks down, the chromosomes condense, centrioles start moving to opposite ends of the pole, and the spindle begins to form. But in this case, in meiosis, it's the homologous chromosomes that pair up, and that's unique to prophase 1. In prophase 1 of mitosis and meiosis, the homologous chromosomes do not form pairs in this way. So again, you're adding highlights, underlining some things, and emphasizing the do not. So homologous chromosomes pair up. This is unique. In metaphase, the spindle fiber is attached to the paired homologous chromosomes, and the pairs line up at the equator. This only occurs in metaphase 1. Okay, so and in this diagram, we're showing the homologous chromosomes as being roughly the same size or so. They carry the same genes. Again, I'm going to explain this a little bit better, but notice that they line up at the equator. In anaphase 1, the chromosomes of each homologous pair start to separate from each other. So see, we're not separating the copies just yet. We're actually getting the homologous chromosomes away from each other, separating them into 2 new cells to be divided a little bit later. And then telophase and cytokinesis, same thing. The spindle breaks down, a new nuclear membrane reforms, the cytoplasm divides, and you get 2 haploid daughter cells. They have a random assortment of chromosomes with one from each homologous pair, and then we're going to move on. Well, the cells will move on into meiosis 2. So meiosis 1 is the separation of the homologous pairs. Now meiosis 2, basically the same process, only this time now we're going to separate the original chromosome and its copy. So prophase 2, same thing, same events. In metaphase 2, the spindle fibers line up at the sister chromatids. In anaphase 2, the sister chromatids separate. And then telophase 2 and cytokinesis, all the other things happen, and now we have 4 haploid cells. So we have 4 cells with half the number of chromosomes as the original. And each new cell has a unique combination of chromosomes. So here is meiosis 2. So again, we've got homologous pairs in separate cells, and then this process separates the original chromosome and its copy so that we've wound up with 4 cells with half the genetic information. So I want you to pause here for just a second and answer the 4 summary questions at the end of the text, and then I'm going to go over those answers. Okay, hopefully you've answered those and you weren't just waiting on me. Okay, so question 1, is the DNA replicated after meiosis 1? That is a big fat no, because we need the genome to be in half. So meiosis will half the genetic information, and then when egg and sperm combine during fertilization, that genetic information is restored into the new offspring. Question 2, describe the main differences between meiosis 1 and meiosis 2. In meiosis 1, it's the homologous pairs that line up at the equator. And in meiosis 2, the sister chromatids line up at the equator. So remember, meiosis 1, the goal is to get the homologs separated from one another. Meiosis 2, the goal is to separate the original chromosome and its copy, the sister chromatids. Question 3, state the phase where each occurs. So sister chromatids are separated in anaphase 2. Homologous chromosomes form pairs in prophase 1. And 2 haploid cells form in telophase 1. Okay, so we really have to think about, again, meiosis 1, separating the homologs, meiosis 2, separating the sister chromatids. And question 4, what's the final product of meiosis? That would be 4 gametes or sex cells with half the number of chromosomes. Okay, so in the chart here, I want to talk a little bit about homologous chromosomes. So you have this diagram and you're going to add in this text that you see. Homologous chromosomes, one is inherited from the male parent and one from the female parent. They're roughly the same size. They carry the same genetic information. And you can see that we've highlighted 5 different genes here. Each one of these represents a gene. And so sometimes each gene can be a different version, and we call that an allele. So you can see here that 2 little r's, 2 big p's. Sorry, let me get that out of the way there. And so there are those different gene pairs. So we describe those as homozygous and heterozygous if you have one of each. And then this is a karyotype to show you that, so like all of our chromosomes are divided up into pairs, typically based on size. And so like this pair 1, these are 2 chromosomes. They're the homologous pairs. Alleles are different versions of the same trait. Okay, so you might have a trait for flower color. The A allele represents purple and the little a allele represents white. So the gene, the trait is flower color. These are the 2 versions. Okay, and so scientists use, letters to designate that stuff. So you could have fur color as a trait. You'd have allele for brown and an allele for chinchilla. Peas are seeds and they come in different colors. They have 2 versions, yellow and green. And then you have this little chart there where we have haploid versus diploid, just to kind of summarize the differences between the 2. And they come from the Greek, the di and hap part are from the Greek prefixes diplos, which means double. So we say that would be the full set of chromosomes, 46. And haploid comes from hapless, which is single, and that would be 23. So again, the 23 are only in sex cells. And then it's just another diagram to kind of show the difference between the 2. Diploid, 2 copies of every chromosome. Haploid, 1 copy of each chromosome. Okay, and then I'm going to ask you again to pause here for a second and answer the what I need to know questions. I'm not going to go over these in this presentation. You'll have access to the key. All right, now that you've answered the what I need to know questions, let's look a little bit about the meiosis and genetic variation. How does meiosis create genetic variation? Well, there's three things that contribute to genetic variation. Fertilization, that's when the egg and sperm combine to make a zygote. And again, you're adding the highlights. I went ahead and just typed everything in for you. I just wanted to kind of go over it. The thing about fertilization is it's totally random. It just depends on which sperm fertilizes the egg first. And if it had been a different sperm that fertilized the egg that became you could be sitting there listening to me with different traits. You could have different hair color, eye color. You could even be the opposite sex. And so random fertilization increases the unique combination of genes. And the chance of getting any one combo from a set of parents is 1 out of over 64 trillion different combinations. So that is incredibly, or creates incredible amounts of genetic variation in offspring. Meiosis contributes to genetic variation through something called independent assortment. That's where the chromosomes line up at the equator in metaphase 1, and they can line up anyway. And they sort out into the sex cells that they're going to become totally randomly. And so this also increases unique combinations of genes. The homologous chromosomes pair up randomly. In human cells, there are about 8 million different combinations that can result. And then finally, something else that's unique to meiosis is called crossing over. This is when homologous chromosomes exchange segments during prophase 1 of meiosis 1. And I'm going to talk about that in just a second a little bit better. But it creates new combinations of genes. And the recombined chromosomes are a combination of genes from both the mother and the father. So let me explain that a little bit better with a diagram. So you start with the homologous chromosomes. They're the same size. They carry the same genes. And then during prophase 1, you can see that the legs of the chromosomes will actually kind of wrap around each other, and they kind of break off and reattach to the other homologous chromosome. And so synapsis is the process of crossing over, and the chiasma is where the homologs cross over. And in this situation here, while crossing over has happened, we call this a tetrad. It's all 4 sister chromatids together. And then finally, we have the recombinant chromatids and the non-recombinant. So the recombinant ones are the ones that have recombined. And so you can see that this chromosome here has a little piece of the red homologous chromosome, and this one has blue. So they've actually switched genes. And so this creates a unique sequence of genes that didn't exist in the previous generation, but does now in the offspring. And the non-recombinant genes have not recombined. So that's crossing over. And I have a link in your doc to this diagram. I just didn't have enough room to kind of keep it in there and keep this Google Doc as short as possible. But just a couple things to kind of go over. Remember, meiosis 1, the object is the separation of homologous pairs of chromosomes. And so independent assortment is shown here. It just depends on which way the chromosomes line up. So you can see here that the 2 blue ones, if you will, are on the same side, but over here they're opposite. So imagine all the pairs that we have, how they can independently assort from one another. And so anaphase 1 and telophase 1, we've separated the homologous chromosomes from one another. And then meiosis 2, we're going to separate the sister chromatids. And so the result is 4 haploid cells with half the genetic information. Now, even when we're here in meiosis 2, these cells are still considered haploid. It's one copy of the chromosome. It just still happens to have its replicated chromosome attached to it. And then here, the haploids and the replications have been separated. Okay, that's it. Thank you for your time and attention.